Thank you. So, our lab is interested in lineage reprogramming, and we specifically study this in the context of hematopoiesis. Yeah, so if you can see here, this is the blood stem cell, which can then branch into either differentiated blood cells. So here on the left, we see the lymphocytes, and here on the right, we see myeloid genes, red blood cells, and so on. So for this talk, I will specifically focus on B cells from the lymphoid lineage, which upon the overexpression of one single transcription factor, which is CBP alpha, we can convert those cells into macrophages. This process, on average, requires one cell cycle. In general, there's no retrodifferentiation, although some myeloid markers, progenitor markers are expressed. So this process was further optimized by using a cell line which expressed CBP alpha fused to an ER domain. So now we have an inducible system. And by the addition of estradiol, you can see that here we start with B cells. This is the cell surface marker CD19. And in the course of 2 to 3 days, we convert B cells into macrophages. And they have been extensively characterized with gene expression analysis. They also eat bacteria and so on. So the question is, how is the macrophage transcriptional program, how is that established in the context of the B cell epigenome? So in order to address this question, we performed chip sequence experiments. So first we probed CBP alpha, PO1 and CBP beta, which are all highly expressed in macrophages. PO1 is also expressed in B cells, but rather at intermediate levels. And we took different time points, and then we probed the binding of those factors. In addition, we also probed for P300 and chromatin marks, which I will show you later. So the bottom line is that in total, we identified about 50,000 non-overlapping CBP alpha binding sites. The majority, though, was transient in nature. So I will focus here on sites, about 8,000 sites that we identified here after 48 hours in our induced macrophages. And in the vicinity of those sites, the genes were merely upregulated, and we did not find enrichment for genes that were downregulated. We further characterized it by motif analysis and Go annotation. And those sites are mainly intergenic, and they are myeloid enhancers. So here you see examples of robust CBP alpha binding. This is the PO1 gene, so it's actually the partners of CBP alpha, and also it's CBP beta. So we see robust binding after 3 hours. But what's also obvious from this slide is that PO1, as you can see here, those sites are actually already pre-bound by PO1 in the B cell epigenome. So this is the non-induced B cells. So in order to get a better picture about what's the starting chromatin configuration of those myeloid enhancers in the context of the B cell epigenome, we looked at the histone 3K4 monomethylation mark and also the K27 acetylation mark. In addition, the K27 trimethylation mark. So here as a reference, you see those 8,000 sites in the induced macrophages. And surprisingly, we see that those myeloid enhancers, the majority of about 2/3, they are already in an active configuration in B cells. So that's what you can see here depicted here. So this is one class, and within this class, about 2/3 is then primed by PO1 as well, and low levels of the K27 trimethylation. On the other hand, we also found a different class, which we here refer to as de novo, which were completely void of those activation marks, but in contrast contained high levels of histone 3K27 trimethylation. So we have two different enhancers in B cells. So we further characterized the starting chromatin configuration by looking at the nucleosome density by using MNA-seq in collaboration with the lab of Michael Beato. And what you can see here on the sites that are primed by PO1 in B cells, you see here in the center of the binding sites, we see a small valley flanked by 2 positioned nucleosomes. So this is the starting configuration. And then upon induction, you see that this gets more pronounced here with a pronounced value. In contrast, what we see here on the de novo sites, and here on top, you see the binding profile of CBP alpha and PO1. But what you can see here is we see very high nucleosome density on those prospective myeloid enhancers. And even when CBP alpha binds here, we have a very high nucleosome density, which then in time, with a delay, resembles the profile as we see here for the other cluster of myeloid enhancers. So next we looked at the chromatin profiles on those two sets of enhancers. And what you can see here, this is what I just showed you before. This is the starting configuration, high levels of P300, K4 monomethylation, K27. And in time you see an increase. And here you actually see a slight decrease a further decrease of K27 trimethylation. But there's virtually none here in the uninduced cells for the de novo set of myeloid enhancers. And also with a delay, for about 48 hours, it reaches a similar level as for the other cluster. So this is here a quantification, what you can see here. So what we also strikingly see is that after induction, the first thing that happens is recruitment of P300 for both sets of myeloid enhancers, but with a different delay. I have to explain this because here this is normalized, this is relative to the uninduced cells, so we subtracted the values here of the uninduced cells. So it's just the value what you see after induction. So increased levels first of P300, then followed by the K4 monomethylation, and finally we see this ordered sequence of events that after 24 hours we see a high level of K27 acetylation. The same is true for the de novo. But the clear difference that we see here is that for the K27 trimethylation, because here there's virtually none, so there is a small reduction, but for the de novo sites, we see this clear reduction after 24 hours. Another very interesting thing actually is that on the primed sites, even though we see a spike in decoration of those chromatin marks, they actually then fall back to some sort of the starting level. And this is not really the case for the de novo ones. So basically what we see here is we have two different kinetics of enhancer activation. And this also relates to differences in gene expression. Primed enhancers are already active in uninduced cells and get further activated. And here with a delay, we see this, with a delay we see the increased expression of the other set of myeloid enhancers. This is the C10 cells, this is the primary B cells. So we come up with a model that we show we have two prospective myeloid enhancers, one that is already primed and CBP alpha acts as a secondary factor, and this is where CBP alpha, what we think, acts as a primary factor or actually can act as a pioneer factor, and then together they upregulate gene expression. So what's the relevance for normal myeloid differentiation? Because this is all done in transdifferentiation. So we took our two set of myeloid enhancers and we followed their activation pattern during normal hematopoiesis. And we did it by using a data set that was recently published. And the striking thing is that the two types of enhancers that we identified in B cells are actually mimicking normal differentiation, in that the primed enhancers are already active in the very early stages, that is here on top of the progenitor cells. They are already active, while the other set, the de novo set of enhancers, they become active at the later stages. So the two different kinetics of myeloid enhancers that we identified during transdifferentiation is reflected during normal differentiation, and other way around. And here you see the expression profile is that even in hematopoietic stem cells, myeloid enhancers are already active, and then they become further active during myeloid differentiation. While the de novo enhancers, they only become active in the transition from GMP to macrophages. So that's from here to here. So what we now have is that we have two different myeloid enhancer pathways that you can see here in green. One type is active in a lot of different hematopoietic cell stages, and the other one only becomes active here at the GMP. And with the transdifferentiation, we kind of short-circuit the two different myeloid pathways. We activate the two different myeloid pathways, and together then, when both pathways are activated, that's when we get functional macrophages. So with this, those are the conclusions, but with this I would like to end. Thank Thomas Graf and the rest of the lab, and I'm happy to take questions. Thank you.